and I've got my web browser open behind that. So I'm going to go ahead and close some of these tabs so I don't get any more confused than I already am. And go back here to the NCBI homepage. And if anybody wants to follow along while I do this, you're welcome to do so. To get to the NCBI homepage, the easiest thing to do is to just search for it in Google if you don't know exactly how to find it. So there are lots of ways to start one of these. I think the simplest thing to do from our perspective is to start at dbVar at the homepage, and I can go here, and you can always grab it from the pull-down list, dbVar. And this, if you're going to any of our Entree database homepages, if you just click search with nothing in the search box, it will usually load the homepage. So here's the dbVar homepage, and I pointed out the various links that are on here already when we did the slides, so I don't think there's anything really more we need to say there. And what we're going to do from here is to link directly into the study browser. And what I'm going to do is we're going to look at this. Actually, I don't really need to do anything if I don't want to, because we can see the 1,000 Genomes Consortium Phase 3 data right here. It was submitted to the European Archive, and so it's got that ESTD accession number. But this page is a useful way of looking at all the studies that are in dbVar and sort them by the various things that would give you the ones that are most useful to you. And any of these things, if I don't see all the different things that are available to filter by, I can just expand this, and you can see all the different organisms, for example, right here. So I can click on human, for example, just to narrow it down. There are no cases, in other words, there are no phenotypes associated with the 1,000 Genomes Project data. This is a control set, so I can filter it by that if I wanted to. If I know the method, and I do that the method there was sequencing, I could pick that, so on and so forth. And I could even, if this has a lot of variants in it, I could filter it by the number of variants. But I can quickly identify the 1,000 Genomes Consortium Phase 3 data, which includes structural variations. So I'm going to go ahead and retrieve this study record. And when we do this, we're essentially in the Entree system, or the way that the record views are showing up in Entree when you do searches. Let me go ahead and get that. So this is a study record. A lot of this talks about mapping, because what happens is these things are submitted on different assemblies. Notice this was submitted on GRCH37, and we've mapped it over to the current assembly, which is GRCH38. So it tells you about the different chromosomes here and how they got over onto the current chromosome record. And if I click here, that will take me into the dbVar genome browser, and I can see that there. We're going to do that in a different way in a little while. If I go over here, you can read about the study up here. Notice the study accession number, what kind of study type it is, and this is the structural variance from the 1,000 genomes population. Of course, you can download those regions, calls, and all those things right here, or go to the FTP site. The sample sets are available here. This particular sample set is interesting. These are basically samples from the Human Genome Project data, so these are going to be records for individual people. And if I click on one of those, it will take me to the biosample database, and I can look at that particular individual person. I'll just go ahead and do that. So this is from the population grade GBR, which is a British from England and Scotland. This first person I happen to pull out is a male, and so this gives you information about the person. And that person's DNA from the cell line that they made from him has been involved in a number of different projects at NCBI. Okay, so what I want to do now is to get the variance in this study. So let me go ahead and follow this link. This is sort of like the links menu that exists on all the Entree database. In this case, in this particular case, it's linking to the subsidiary variant records that are part of this study. These are going to be the variant regions. And so really what they did was it did a real simple search for ESTD214. Just to show you that this is an Entree database, there are fielded queries that I can do here. In this particular case, it really doesn't make any difference, but I could use the study accession field if I want to, and I should get exactly the same results. I do. And the other thing I would want to do is to add something to this search. I could do that simply by typing it, if I want to, I can use the advanced interface just to point out to you that that does work, just like it does in all other Entree databases. I can go to advanced, and I can look at the various fields that are available here. One obvious field that's interesting is gene name. And what I'm looking for is glutathione transferase M1, which is the name of that gene, which is one of the mu-class glutathione transferases. So I can take that and I can add my search for the study accession here and run the search to get my results. And I get 3 different variant regions. What's interesting about them is that one of them actually affects 2 of the mu-class glutathione transferases. It overlaps with the M1 gene and the M2 gene, which, as we'll see, are right next to each other on chromosome 1. And then there are 2 others that represent changes at the level of the GST M1 gene. So I'm going to go ahead and retrieve the region ID here for the one that affects both M1 and M2. And notice that there's a heck of a lot of variant calls here, and that's going to cause a little bit of an issue when we look at this in the graphical sequence viewer, because there's going to be a lot of them. We can fix that problem pretty easily, and it's a good example of how to modify track settings in the graphical sequence viewer. So that should take a second to load. There we go. Okay. So here's a copy number variation that tells you about the method. There's no clinical assertion. It's a region that's about 22,000 bases long, so that's not a terribly large region. And that's where it is. It's on the P arm of chromosome 1, not too far away from the centromere. Here's the track. This is the dbVar Genomes Consortium Phase 3 data. This is our study accession. There's a heck of a lot of these guys here. So what I can do to make my life a little bit easier is to sort of change the track settings here to make this a little bit easier to look at. So I'm going to do that first. I'm going to put my mouse pointer over here. If I mouse over a track in the graphical sequence viewer, I get a little pop-up that has these, this little gear icon. That's essentially the same as going to the configure menu up there on the right. It's just a little bit of a shortcut, so it's just going to allow me to configure this one particular track. So I'm going to do that. So right now we're seeing everything. What would be nice is to sort of collapse these variant calls into a single graphic. So that's what this show parent and merge children will do. So I can click that and I can click apply. So that makes my display a little bit easier to look at, and it gives me a nice overview of what's going on here. So these are all the other variant regions from that particular study that are on this part of chromosome 1. And you can see the GST-M1 gene here, and it's been completely affected by these, by this variant region and the underlying variant calls here. And then there are some other smaller ones that affect it too. Now, in order to see what these various colors in the graphics mean, let me zoom out a little bit too, just to point out to you where that GST-M2 gene is. Let's expand this a little bit. So you can see the gene right before it on chromosome 1, and there's several of these mu-class glutathione transferases that are part of a tandem duplication event through here. So the one we're looking at, the one that's highlighted here, affects the 3' end of GST mu2 as well as the entire GST-M1 gene. So another thing that's useful to be able to do is to sort of understand what the graphics are representing here. And as I told you, when we were looking at the slides, red means that it's been a deletion, and the blue means that there's been an increase in copy number. But you can see that for yourself by just going to this little question mark here. This is the track legend. And so if you ever want to know what these various things mean, this is the copy, the blue is a copy number gain, and the sort of lighter area says that there is a known length of deletion, but it varies between the various variant calls, and so on and so forth. And you can look at the various meanings of these different kinds of graphics that we have here. Okay, so we're back here looking at this particular region in the viewer. So we know that what we're looking at here for that particular variant region is a copy number loss. So these individuals are missing the GSTM1 gene plus a part of the GSTM2 gene. And there's a whole bunch of these mu-class glutathione S transferases there on chromosome 1. The phenotypic consequences of missing these is basically unknown, but there's some indication that when you're missing glutathione S transferases, you might be more susceptible to certain kinds of toxicants. What's interesting to do here, we don't have available the genotype information here, but it is available in the 1,000 genomes data. As I said, there are no clinical assertions associated with this. There was no validation information, but if those things existed, they would be present here. This is the same kind of thing we saw before, where we were looking at those individuals from the 1,000 Genomes Project data. But notice that I can see which individuals are homozygous or heterozygous at this particular position with a copy number variation. I'd have to look at the data to see exactly which way that went, in terms of if they were homozygous for the deletion or homozygous for a copy number increase. Okay, so what I want to do now is to go back and get that particular region displayed into the dbVar genome browser. And one way I can do that is to go just back to the dbVar homepage. If I had done this the way that I planned to do it, I would have opened this in a new tab, but I didn't. I could hit my back button a bunch of times. I could just go here. I could go to the advanced interface here on dbVar. And to retrieve these records here. Okay, so now what I want to do is I'm going to follow the link from that result and load this into the dbVar genome browser on human genome build 38. This is a position on chromosome, a region on chromosome 1. So when I come into the browser, it tells me about where I am on the chromosome, and we can see basically the same kinds of things we were seeing in the graphical sequence viewer before. Notice that by default, the display on that particular track is to show me the collapsed view of the variant regions and variant calls, which is helpful. So I can change my assembly. I can search here. We're going to do that in a little bit later on. I could upload my own tracks if I want to, just to show you that that's possible. I have some files here on the FTP site. Let me go ahead and see it. We'll see if this displays. We'll give it a shot. Okay. So this is an example of a GVF file that you could have made yourself from your own data, and we could load that into the browser if we want to. So we'll go ahead and do that now. I have that on my desktop. So I can go ahead and add my own data here. This is it here, thanks. So there's my own track there, the variant calls that I put in the data, and they're shown up here as a track. So you can upload your own tracks if you want to. We won't spend a lot of time with this today, but I just wanted to show you that you can do that. If I want to get rid of them, which I will do, I can simply click this minus sign here, and it says, do you really want to remove this track? And I'll just say OK. And what I want to do now is to add some additional tracks to this region to look for maybe some pathogenic variants and things like that. So let's take a look at the entire region containing the glutathione, the new class glutathione S transferases on chromosome 1. What I can do here is zoom out a little bit. And here they are. So there's a mu4, a mu2, a mu1, a mu5, a mu3. They're in no particular order in terms of their names, but this is how they are organized on chromosome 1. And I've picked up some additional variations that are in the 1,000 Genomes Project data, as you can see as well when I did that. And what I can do down here is I can add tracks if I want to. So here's the track that's currently being displayed, which is the 1,000 Genomes Project data. These are just showing you the study accession numbers here. Notice that this is an E, so that was submitted to the European Archive. If I expand this, I now can read about them, which is helpful. So I can see what kinds of data are available. I can filter them here, which is also useful to be able to do. And so one of the things I can do is to get rid of everything here except for the case set, because they're likely to have clinical assertions associated with them. And I can go ahead and add those if I want to. So I'm going to take two of the ClinGen studies here. This one here, which has 3 variant calls in the region. And notice what it's displaying for me is the ones that actually do have data in that region. So some of these things, for example, in this particular case, I don't have any of the curated data sets available for this region. I can go ahead and add this one. And I can add the other one down here in a TD101. There it goes. Okay. So notice that in these particular studies, the ClinGen ones that we've looked at, it classified the variants into various categories in terms of their pathogenicity, likely pathogenic, other, uncertain significance, benign, likely benign, pathogenic. And so we have some pathogenic variants in here. There are copy number increases and there are copy number decreases. This happens to be one. If I mouse over any of the variant calls, it's going to tell me about the phenotypes that are associated with that particular individual who had this particular problem. And so this particular one that I've picked from the NSTD37 study, this says it has 2 phenotypes listed, autistic behavior and intellectual disability. And so I can go ahead and look at that if I want to. Just another tip for the graphical sequence viewer. If you don't want that little box to go away, you can pin it down with that safety pin, with that push pin thing there. I can go ahead and open this one in dbVar. Yeah, it's right here. And so there are a number of other variants that are in this region too, and that's interesting. We didn't see them when we looked at the the dbVar genome browser. Well, the reason is because this copy number increase is a huge region that incorporates 15.6 million bases of chromosome 1. And in fact, this is a thing that you could see on a karyotype. This person has an extra ring chromosome in their karyotype, and that ring chromosome is made from this region of chromosome 1. And so this is a very large-scale variation in this person's genome that's associated with some pathogenicity. And if you want to see that kind of information, you can look over here at the clinical assertions. This is copy number gain. There is the link to the ClinVar ID, which would be the original source of this particular variant. And there are 2 phenotypes associated with this, both of which have links to MedGen. And I can go ahead and just visit this one, the autistic behavior one. So this is a concept ID that has to do with what's called autism spectrum disorder. And this happens to be the phenotype that they assign to this particular individual. This is the MedGen record, which aggregates data from a number of sources and has controlled vocabularies for classifying these phenotypes, which makes it pretty useful for finding things. These literature citations are created sort of semi-automatically to keep these records up to date. Okay, so what I want to do now is sort of shift gears a little bit. We're going to now do a search within the dbVar genome browser. And I'm going to look for a particular phenotype. I could also look for a particular gene or other things that I want to. This happens to be, I know that I'm going to find a gene that's on this chromosome, which makes it a little bit easier to sort of navigate for the purposes of this example. And I'll go ahead and do that. So let me go ahead and put my phenotype up here. And the phenotype that I'm looking for is cleft palate. So I get a number of results. The one, of course, that I'm going to take you to is the IRF6 gene, which is on the same chromosome on the other side of the centromere. So this ClinGen study has some information in here that has some pathogenic variants. Actually, what I want to do is to load a different set of studies. So let me go ahead and open this track here, and I'm going to get rid of the case set, go to the curated stuff here. And there are two of these curated studies. One of them is this ClinGen curated dosage sensitivity map study. And the other one, these are various kinds of variants coming from things like OMEM or from ClinVar or from gene reviews that basically our ClinVar group has put together. And so these talk about basically phenotypes that are associated with the deletions of a particular gene. So I'm going to go ahead and add those two. And I can get rid of these other ones because we don't need to see those anymore. And I left the 1,000 Genome Project data up there just for interest. Notice that there's nothing in that region. The reason that there's nothing in that region is that if you had something in that region, you might be in a heap of trouble, because this causes pathology. So there are no variants, there are no large-scale variants from 1,000 Genomes Project in there. And these are the curated variants here. If I put my mouse pointer over one of the variant calls, this is the phenotype we talked about, which is van der Waals syndrome. Pathogenic, of course. And here's another one here for van der Waals syndrome here. I'll go ahead and retrieve this one here, which is the one from the ClinGen study. Let me go ahead and retrieve that. So this basically is just going to be about mapping them. There's no validation information available for this one. But what there is a clinical assertion, and it has a link to the van der Waals syndrome page in MedGen, and then links to other resources. And this, of course, is the same one that I showed you in the slide earlier today. So I did this particular search using the dbVar genome browser. You could do exactly the same thing using the Entree system if you wanted to find a particular variant. Okay, so this is the end of my presentation. And so what I'd like to do now is take a look at what questions we have. We'll definitely have them up in a document after the webinar. If you think of anything else and you want to talk to us about it, please write to us at webinars@ncbi.nlm.nih.gov, or you can write to the help desk and it will get to us. So that's it for today. I'm going to go ahead and end the webinar, and we'll see you next time.